Hello students, I hope you all are doing well. Well, in this video lecture, we are going to continue further with the topic reproduction in flowering plants. And today we are going to see the development of embryo, okay? Or we can say the embryogenesis in flowering plants, okay? Well, to start with the development of embryogenesis, let's first draw an ovule, okay? As in the previous class, when we were studying the development of endosperm, we saw the most common type of endosperm is nuclear endosperm. Okay, so here I'm taking the most common case and we'll start from here. So this is a typical ovule after fertilization with the development of or developed nuclear endosperm. Okay, now what we see here is still the zygote is not developing, right? We spoke about this, that the zygote undergo a resting phase, whereas the PEN or primary endosperm nucleus start to develop immediately. Okay, and that's why in plants the formation or the development of endosperm precedes the development of embryo. Okay, great. So in most of the plants, the embryo development from zygote starts only when a considerable amount of endosperm has been formed. Why it is so? It is so that it can be ensured that the developing embryo will never be having the shortage of food. Okay, so to ensure the nutrient supply, this is done. Okay, that is the PEN or endosperm development precedes the development of embryo, and this is only to make sure the nutrition of the developing embryo. Okay, so once this endosperm or considerable amount of endosperm is formed, then the zygote starts to develop into embryo, right? So see, now embryogenesis occurs, and just to give you a glimpse, what I'm going to do here is I'm going to draw a matured embryo, right? So see, this zygote has now developed into this full grown embryo, which is shown in pink here. Okay, now see, this is embryo. Okay, and these are the two cotyledons. Okay, so the single-celled zygote has developed into a multicellular embryo having two cotyledons here. So typically this is the case of a dicotyledon embryo, which I'm taking or which we are studying here. Okay, the remaining dots shows the free endosperm, yet this embryo has endosperms. Okay, great. So can you see, this is embryogenesis. A zygote has developed into this multicellular embryo. Now let us see how this happened, how this one-celled or single-celled zygote develops into this full-grown embryo. Okay, what are the different stages in it, how this divides, how they form it. Okay, so we are again starting with the single-cell zygote. Okay, so this is zygote here, and first of all, the zygote undergo a transverse division to form two cells. Okay, transverse mitosis to form two cells. Okay, now let me tell you what I am not describing here is this first mitotic division in the zygote can either be complete or incomplete. Okay, that is, if this is complete, then there will be two cells formed. Okay, if it is incomplete, then also two cells will be formed, but they will not be distinct. They will not be totally different from each other. Okay, great. Now if complete division occurs, the first division in the zygote is complete, then we call it holoblastic. If not, then we call it meroblastic. but this is something you do not have to remember, okay, just for knowledge. Okay, great. So here we are taking the holoblastic development. The zygote, the cell of or the zygote undergo a transverse complete mitotic division. As a result, two cells are formed. Okay, the lower cell, that is the cell at the micropylar side, okay, remember it is very much important, the position is important, okay, the cell on the micropylar side, which I have made in white, and the cell on the other side, that is the chalazial side, I have drawn in pink. Okay, now see what happens. This lower cell is known as basal cell or suppressor cell, sorry, basal cell or suspensor cell. Okay, why it is so? Because this cell is going to make the suspensor of the embryo. Okay, so they are going to make the suspensor. That's why the lower cell is called basal or suspensor cell. Great. The upper cell is called apical or embryonal cell. Okay, now there is a third name, it is also known as terminal cell. Okay, apical or terminal or embryonal cell. Okay, now why we are calling it embryonal cell? Because this particular cell is going to make the whole embryo. Okay, so see, the embryo has mainly two parts. One is called suspensor and the other one is called embryo, embryonal axis, right? So actual embryo is made by embryonal cell. The basal or suspensor cell is just going to make the suspensor, okay? And this embryo is, sorry, the suspensor is not the part of the embryo, okay? So it is just a correction. So basal cell and the apical cell or embryonal cell. Now what happens is the basal cell, okay, enlarges and undergo again transverse divisions to form 6 to 10 celled suspensor. Okay, so these are the suspensors. Okay, this is suspensor cells. These are formed from the suspensor cell through the transverse mitotic division. Okay, transverse. Great. At the same time, what happens? The apical cell or the embryonal cell, okay, divides vertically. It is not going to divide transverse. The first division in the apical or embryonal cell is vertical to form two cells. Okay, see, now this two-celled stage is called a proembryo. This is called a proembryo. याद रखिएगा इस बात को. This is proembryo. Now vertical division हुआ यहाँ पे. इतना डिटेल में आपको याद रखने की जरूरत नहीं है कि वर्टिकल हुआ नहीं हुआ, बस आपको ये याद जरूर रखना है कि प्रो एम्ब्रियो डेवलप्स फ्रॉम द एपिकल और एम्ब्रियोनल सेल इन केस ऑफ होलोब्लास्टिक डिवीजन. ओके, होलोब्लास्टिक डेवलपमेंट. ठीक है, अब क्या होता है, इसके बाद से ये जो दो सेल्ड स्टेज है आपका प्रो एम्ब्रियो है, इसमें अगेन डिवीजन होगा टू फॉर्म फोर सेल्ड स्टेज, एंड देन द फोर सेल स्टेज में फिर से डिवीजन होगा टू फॉर्म एट सेल्ड स्टेज. ओके, ये सब मैं लिख नहीं रहा हूं, सिर्फ सुनते रहिए. दिस इज नॉट very much important for you. However, it will help you to picturize the whole concept, the whole process of embryogenesis. That's why we are discussing it here. Okay, so it is just for your understanding part. From examination point of view, it is not that important. Whatever I am just verbally telling you, which is not being written on the screen. Okay, now listen, these two cells will again divide to form four cell, and then four cell will form the eight celled stage. Out of these eight cells, the chalazial end four cells will form the plumal axis, and the lower will make the radical. Okay, now in these eight cells, again there are two rounds of periclinal division. That division will be at an angle, so making peripheral cells. Okay, out of that, the outer cells makes the epidermal tissue that will make the epidermis. The inner cell will make the periplasma that will form the stele and the cortex. Okay, now this you can relate from the quiescent theory. or from the anatomy what we have already learned in class 11. Okay, great. So these all things are formed in the embryo from these apical cell. Okay, so let's see now. So this is dividing. So what I'm drawing first is the periclinal part, the outer part. Okay, and then I'm just making it, and then inner there are cells. Well, this is not the actual arrangement of these cells, but that is okay because you do not have to study them. You just remember that this stage is called globular embryo. Okay, now from the apical embryo forms the proembryo, from proembryo forms the globular embryo because it appears spherical in shape, that's why it is globular. And this stalk like thing is called suspensor cells or suspensor, which is made up of 6 to 10 cells. The last cell of the suspensor, which is larger in size, is called haustral cell. Now, why we call it haustral or haustorial cell? Because this cell is haustorical in nature, as in this will degrade the surrounding tissue or surrounding cells and take up the nutrients from them, and this nutrients will be given to the developing embryo. That's why it is haustorial in nature. Correct? You can recall from haustoria, from the parasitic plants. Okay, so it is haustorial in nature. Great. Now what happens? This whole structure is called this pink circular or spherical structure is called globular embryo, and then there is a suspensor. Now what happens is further there is development and the suspensor remains like that, or it may grow a little bit more. Okay, now when the suspensor grows, okay, this pushes the growing embryo further into the endosperm, or it pushes it towards the chaliceal end into the endosperm. Okay, that's why it is endoscopic movement. Why it is done so? It is done so that the growing embryo is surrounded by the endosperm, which will provide it nutrition. So again, it is a mechanism to ensure that ample amount of nutrition is being given to the growing embryo, or growing embryo will never be short of nutrition. Okay, so do remember what it is doing. The suspensor is pushing the embryo deep into the endosperm. Okay, great. So this will continue. Suspensor may grow, may not grow, but the globular embryo now will show some changes. It will further grow to take this heart shape. Okay, to take this heart shape, and this stage is called heart shaped embryo. Okay, now you'll ask, what are these two notches? So these two notches coming up are the precursor of cotyledons. Okay, so these are the two structures that are going to grow to form the cotyledons, and these two notches give the whole structure appearance of a heart shape, and that's why it is called heart shaped embryo. Okay, great. Now once we have this heart-shaped embryo, this heart-shaped embryo further develops. Okay, so these two notches again develops to form fully formed cotyledon. Okay, can you see here? So these are all cells, and these two structures further developed into the two cotyledons. Okay, great. So this is the structure of a fully developed embryo. Now, in case of capsella, okay, because of the structure of the whole seed, what happens is the embryo bends a little bit. okay, the developing embryo bends and give it a typical structure of horseshoe shape, or it appears like a torpedo, and that is called horseshoe shaped embryo or torpedo embryo. Even this developed or matured embryo is also known as torpedo shaped or torpedo embryo. Okay, now these two are cotyledons and the central part is embryonal axis. So are you getting it? Can you see it how? Let me. Okay, can you see how this single cell zygote has finally developed into this fully developed embryo? The shape, how the shape changes, how the cell divide. Okay, great. So this is the whole process of embryogenesis. Okay, now let's draw a fully developed embryo or a matured embryo in detail to understand what are different structures within an embryo. Okay, and for that matter, here we are again taking a dicot embryo. Okay, so let's draw. So see, what I am drawing in pink is the embryo. Okay, the matured embryo structure. What I'm drawing here in blue are the cotyledons. Okay, so there are these two cotyledons, and the place where the two cotyledons are attached, that is in the middle part of this embryonal axis. Okay, so this pink part is embryonal axis, is called cotyledonary axis. Okay, or tigellum. Let me draw it and then we will mark it. So these two are cotyledons, and this middle portion where these cotyledons are attached is called cotyledonary axis or embryonal axis or tigellum. These all are the same thing. Okay, now the tissue just above this cotyledonary axis is called epicotyle. This is called epicotyle. Why so? Because ep means above. So above the cotyledonary axis is epicotyle. Similarly, below the cotyledonary axis is hypocotyle. Okay. Now, above the epicotyle, what you find is plumule and then this coleoptile. Okay. So plumule and coleoptile are found in epicotyle region. Now, in or below the hypocotyle, what you'll find is coleorhiza. Okay, coleorhiza, the root cap tissue and the radicle. So root cap and radicle, correct? So what you can see here is in a matured embryo, you find cotyledonary axis through which or from where the two cotyledons remain attached, and then above that epicotyle, and uh, above epicotyle you find coleoptile and plumule. Similarly, below the cotyledonary axis you find hypocotyle, and below the hypocotyle you are going to find radicle, root cap tissue and coleorhiza. So this is a detailed structure of dicot embryo, matured embryo. Okay, now let's see a monocot embryo. If we have to study the monocot embryo, the best or the most studied embryo is the embryo of maize or corn embryo. Okay, now this particular picture I have taken from NCERT. You all have seen it or you must have read this. What we can see here in a monocot embryo, there is a single cotyledon, right? That's why it is monocot, and this single cotyledon is in the shape of a shield. Okay, it's a triangular shield shaped. Okay, and this shield shaped triangular single cotyledon is called scutellum. Do remember this. There has been question frequently asking about scutellum. Okay, so scutellum is single shield shaped cotyledon found in monocot embryo. Great. Now this cotyledon is attached to the embryonal axis at the middle point, and this point is called embryonal axis or tigellum. It is similar to the dicot embryo that we have seen. Okay, now above to this embryonal axis contains the structure that are going to make shoot. Okay, now below this embryonal axis are the structures that are responsible for making root or root system, shoot system and root system respectively. Okay, now one more thing to observe here is this thing called EP blast, correct? Now EP blast is a small flap like structure originating from the monocot embryo. Now what is it? This is actually the remains of the second cotyledon. Now why does it come here? Listen, monocot plants have been developed from dicot plants. Okay, so monocot plants are in many things advanced than dicot plants, and they have developed from the dicot plants. So when they developed, they did a lot of changes. However, in case of embryo, they still have this remain of second cotyledon, and this rudimentary or remain of the second cotyledon is called epiblast. Okay, great. Now when we go in the epicotyle region, okay, yani ki above this axis, we find that there is coleopatile, and inner to the coleopatile is shoot apex, which is also known as plumule. Okay, now when we move downwards down to the embryonal axis or tigellum, what we see is again coleorhiza, root cap and radicle, so similar to the dicot embryo. Okay, so this is how a monocot embryo looks like. These are the structures of monocot embryo. Just take care of scutellum. Okay, it is important. Well, now we have seen the embryogenesis or the development of embryo, both the dicot embryo and the monocot embryo. Let's see some other terms which are being frequently asked now. The development of embryo was discovered by Hanston. Okay, Hanston, and this was in case of this was in case of Capsella plant. Okay, so the development of embryo first of all was discovered or reported or studied by Hanston in case of Capsella plant. Okay, this is a Capsella bursa pastoris. Now, the second thing is the study of development of embryo is called embryology. Is called embryology. याद रखिएगा इसको. Embryo के development की study is embryology. Now, next thing is very important. Panchanan Maheshwari. Do remember, an Indian botanist, Indian scientist. He is a very recognized scientist. He worked on the embryology of plants, and his work is a milestone. Okay, he was the person who discovered different type of embryology, okay, or embryogeny. He reported 5 types of embryogenesis. Okay, what are they? Now, first one is crucifer type. Okay, now the crucifer type of embryogenesis is typical to crucifery family and a lot of plant families. So it is the most common type of embryogenesis, and this is the type that we have studied in this lecture. Okay, so whatever we have studied about a dicot embryogenesis was actually a crucifer type embryogenesis. Okay, now apart from that, there are others as well. Now there are 4 others. The second one is called asterad type, or third one is called solanad type, chirophyllid type, and the fifth one is called chenopodiad type. Okay, now this is just, I'm telling you for your knowledge. From exam point of view, it is not that important, so I would not advise you to mug it up or to devote a lot of time on it. Just have an understanding there that there are 5 different types of embryogenesis. Okay, and the one you studied is not the only type how embryo development occurs in plants. Okay, it is the most common type, but it is not the only type. Okay, apart from that, there is asterad, solanad, Chirophylla and Chenopodea. Okay, so these are different types of embryogenesis. Okay, so first one, crucifer type, is what we have to study. Rest 4 is not very much important for us. So that's it. This is how embryogenesis occurs. This is how embryo develops in a plant. Okay. So what all we have seen up till here is how after fertilization the PEN is formed and zygote is formed. Then we saw how after this double fertilization the PEN develops into endosperm. What are different types of endosperm, right? We saw nuclear, we saw cellular, we saw halovial, we saw ruminant, right? And then we saw in this video lecture how the zygote develops into a fully matured embryo. Okay, so this is it for this video lecture. In next video lectures we will discuss about the seed formation, okay, and fruit and the remaining topics of this particular chapter. We are, closing towards finishing this chapter. So stay tuned and stay safe. Thank you.